Hello cellmates, we're back with some more biology. So today we're going to do the cell cycle and its checkpoints, which we'll just talk about as we get to them. But we'll start with interphase, and in interphase it's composed of three parts. The first one is G1. Oh, my laptop just died, so I have to do this from memory. Let's go. The first phase is G1, in which the cell is making sure that conditions are favorable, that the DNA that it already has is not damaged, and if this is good, then it's good to move on to the next phase. But G1 is known as like first growth, so that's where it all begins. And in the next phase, it's symbolized by the letter S, and S is for synthesis, which is building up. It's making more DNA, because eventually the cell will need to split into two, so you want to make sure that it has enough DNA to do that, and that they all have equal parts. So during this phase, we're just making more DNA and replicating that, and then we are good to move on to G2, in which the cell is once again making sure that now with all its new DNA, that none of it is damaged, just making sure there's enough proteins and that everything is there to move on into the next phase, and that is also a checkpoint. So at the beginning of interphase, at the end of interphase, checkpoints. And then we move on to mitosis, which is represented by the acronym PMAT. I'm sure you heard it. So first we have prophase, and in prophase we are able to make sure that the spindle fibers begin to form. So the spindle fibers are that thing that will eventually pull our sister chromatids apart, but it's first making sure that the spindle fibers are forming, and also that the chromosomes that are there are tightly wound, so that everything's starting to get ready. And then we move on to prometaphase, and in prometaphase, I had to peek. I had to make sure I was telling you the right thing, so my laptop is not plugged in. But in prometaphase, this is when the spindle fibers are finally sending out their fishing lines and attaching to the chromosomes. So important part. And once they're attached, we're going to move on to metaphase, which is making sure that the chromosomes are all lined up exactly in the middle, which is also our last checkpoint. And then if everything's exactly in the middle, everything's where it should be, and everything's attached, then we can move on to anaphase, in which the sister chromatids that are making up this chromosome are splitting apart and moving to opposite sides of the cell there in different poles. And we can end with telophase, in which everything has been split apart to different size, and a new nuclear envelope will start to form. And in cytokinesis, which is that very last thing, is where the cytoplasm is split, and they're truly two separate daughter cells. And it's also important to note that these daughter cells are diploid, so there's 23 pairs of chromosomes, so like 46 total. And that's all I have today.